We are welcoming back Dr. Christine Metz. She has been doing some incredible, innovative work in the lab, trying to figure a non-invasive way to diagnose endometriosis. This is groundbreaking. Dr. Metz, can you give a little background about who you are and what you do?
Sure. Thank you so much for inviting me and hosting me today. I'm excited to share the research that we've been doing through the ROSE study, which stands for Research Outsmarts Endometriosis.
Love that.
And a little bit about me. I'm a biomedical researcher here at the Feinstein Institutes for Medical Research at Northwell Health. We are located just 20 miles from New York City on Long Island. And I have spent probably the last half of my career totally focused on women's health, and my first half of my career focused on inflammation. And I applied what I learned from my first half of my career to women's health and became interested in studying endometriosis in the late 1990s, early 2000s. I was actually introduced to it by Dr. Ali Akoum, who was in Quebec at the time and was very interested in the inflammatory components of endometriosis. And luckily, here at the Feinstein Institutes, we were given a grant, a seed grant from the Endometriosis Foundation of America that allowed us to really delve into studying endometriosis. And that really started about a decade ago in 2013. And without that seed money, we would never have gotten to where we are now. And our goals at the time that we received that seed funding from the EFA and founded the ROSE study together with Dr. Peter Gregerson, we came up with goals for the study. And our first goal was to really understand the development of endometriosis. What is it? What is this condition, and how does it progress? And what are the various subtypes of this condition? And we felt that if we understood those fundamental things, we could better develop a non-invasive diagnostic. And hopefully that would lead to new treatments. And the reason that we were very interested in developing a non-invasive diagnostic is that the condition of endometriosis has been long time plagued with very long delays in the time of patients experiencing symptoms to the time that they're being diagnosed. And on average, when we started our study, it was between 7 to 12 years between the time of symptoms and the time of diagnosis, which we thought was extremely frustrating, both for the patients and for the treating physicians. And we tried to understand why that delay exists. And there are many reasons, and probably the most obvious is that the dismissal of pain in women, and particularly young women and adolescent girls, is a problem in our society. And that has been a problem for a long time. And it has been shown in many emergency room studies that women's pain is more likely to be dismissed. And then in addition to that, there are many other reasons that there are delays in the diagnosis. Everything from misconceptions about the disease and gaps in knowledge that kind of delay referrals for people who are seeing their gynecologist, to making sure that they see laparoscopic surgeons. It's a really complex disease. Symptoms are vague, they're nonspecific. The extent of disease may not reflect the symptoms that patients have, making it very difficult to diagnose. And in some patients, they may have very mild or almost no symptoms, and then they could be diagnosed with endometriosis when they confront infertility. And of course, I think the gorilla in the room is that the current definitive diagnosis for endometriosis is that it requires laparoscopic surgery. And many teens and adults delay that surgery. So there was clearly a need to develop an early non-invasive diagnostic for endometriosis. And that's what we kind of tried to set out to do a decade ago. And we kind of took an interesting twist in that we decided to study menstrual blood and use that as the basis for the diagnostic. So, the question is, why on earth would menstrual blood be indicative of endometriosis, right? That's the big question that we get. But it's very clear that endometriosis lesions look under a microscope like the cells that come from the endometrium. The endometrium is the lining of the uterus. Now, every single month, teens and people with uteruses bleed in response to their hormones. And the uterus is not a closed system. So during menstruation, menses is actually pumped into the pelvic cavity. And that's where about 85% of patients have lesions. So it's very clear that it contributes to the development of endometriosis. Not to say that there are many other factors that could contribute to endometriosis, but clearly menstrual blood is one of them. Now, one could say, okay, most people menstruate into their pelvic cavity, but why do only a few people get this condition? And our hypothesis is one that is based in some scientific findings that the lining of the uterus of people with endometriosis, or those who are predisposed to get endometriosis, is different when compared to unaffected healthy controls. And there's a lot of scientific literature supporting that observation. The main differences are related to inflammation and a defect in the cellular response to a differentiation pathway that's required for pregnancy. So that's very specific to humans, and only menstruating animals have that event. So most species in the world do not menstruate, and they do not get endometriosis. There's only 4% of animals that actually menstruate, and endometriosis has only been identified in those species. So it seems very like a good link to be studying as a potential diagnostic tool, as well as a biological sample to understand the disease. So we've been collecting menstrual blood for about 10 years. And I will say that the first 2 years were spent just on trying to understand how to collect it from patients across North America, how they would ship it, how we would process it, and different techniques for analyzing it. Because menstrual blood prior to our studies was not studied in the context of a disease such as endometriosis. It had not been well established in the literature on how to study it. Most people studied the endometrium by doing surgical biopsies of the endometrium and taking those tissues to work with. So this approach, which includes the shed endometrium or the lining of the uterus, was a totally new one. And so for us, the first couple of years, it was really just painstaking work to establish how we would process it and work with the materials. And luckily for us, we identified that there were differences in particular cells found in menstrual blood that were similar findings to what had already been reported with invasive biopsies. So this differentiation defect that I was talking about, we replicated that we could actually see it in the menstrual blood. And we thought at that time, oh, wow, we've got it. We have a diagnostic for endometriosis. But we learned pretty quickly that it was relatively nonspecific and that it was going to be very costly to collect cells and grow them and do these downstream kind of complicated analyses. And lucky for us, there was a new emerging technology that we call single cell RNA sequencing that became available. And what this approach allows us to do is to take a sample of menstrual blood, collect the endometrial tissue or the tissue lining the uterus that's there, and subject it to an analysis that we can define the gene expression patterns in all the various cells that are there. And what we have done is shown very clearly that there are specific gene expression patterns and cell types that are different when we study menstrual blood from a patient with endometriosis versus a patient without endometriosis. In the case of what's different between patients with endometriosis versus without, we see evidence that there's increased inflammation in endometriosis. So we have higher numbers of B cells, okay? And that kind of indicates that there's an inflammatory process there that is beyond what's found in the control samples. The other thing that we see are gene expression patterns in a group of cells called stromal cells that are important for pregnancy and fertility that also show a pro-inflammatory phenotype or characteristic, as well as a pro-fibrotic characteristic. And we believe that's very important, and it may tell us something about the disease progression in people with endometriosis. And on the side of the controls, we see that they have many more something called uterine natural killer cells than endometriosis patients. And those are critical for fertility. And so perhaps this may be one of the implications that's leading to infertility in patients with endo. And the last difference is this increased differentiation of stromal cells in controls when compared to patients with endometriosis. So it's very clear that there are numerous distinguishing characteristic features in endometriosis menstrual effluent compared to menstrual effluent from healthy controls. But we want to be able to detect that in people who are symptomatic, right? It's great to say you see these differences in patients who have already been diagnosed, but the most important is to show that we can replicate this in symptomatic patients. So currently we have an ongoing clinical trial in which we are verifying our findings that we have from our previous work that have been published, showing the differences between healthy controls and endometriosis patients with respect to their menstrual effluent. And we are enrolling again healthy controls, people without any symptoms, and enrolling people who are symptomatic, who are actually seeing a laparoscopic surgeon as part of their standard of care. They've chosen to have surgery, or they're pursuing surgery as a diagnostic tool. And we collect menstrual blood from those patients prior to their surgery. And then we'll compare the results after they get their diagnostic pathology report back after the laparoscopic surgery. And that study is about midway. We've enrolled about half of the subjects so far. We have done a small interim analysis several months ago, and it looks very promising, and that's why the study is continuing. And it's really a matter of getting enough participants who are thinking about surgery and can provide a menstrual effluent sample for us to get results so that we can compile our results and bring them to the FDA. So our hope is by this fall, at some point, we would put all of our data together to present to the FDA to request permission to have a larger study in which we would replicate what we found in a much larger population to really demonstrate that we have a screening tool. And that's really what we're trying to develop here, is a screening tool to identify those people who are really at risk for developing endometriosis and likely should undergo the laparoscopic surgery. I think to go right from menstrual effluent to a diagnostic is a big leap, because our societies in the United States that define endometriosis have defined it on their lesions. So clearly, the assessment of menstrual effluent is not a direct assessment of a lesion. And we know that patients with more severe cases of endometriosis benefit from laparoscopic surgery to remove those lesions. So there is still a strong component to support laparoscopic surgery in patients with endo. We are working with many physicians here at Northwell Health who refer patients who they think meet criteria to participate in the study. And we also get people from online, and people can Google Rose and endometriosis, and they will find the Feinstein study. It'll take you right to the website. And we enroll participants in what we call Rose 1 or Original Rose, which everybody is invited to participate in, healthy controls, people who are symptomatic, as well as people who are diagnosed with endometriosis. And right now in our registry, we have over 2,600 subjects. Wow. And then we have the Rose 2 study, which is specific for patients who are thinking about laparoscopic surgery and are actually seeing a laparoscopic surgeon to undergo diagnosis. And they can be enrolled into the Rose 2 study. But both studies are on our website at Northwell. So if you Google Rose and endometriosis, they'll find. We are depending upon laparoscopic surgery as a definitive diagnostic tool. And I can tell you from our personal experience with the Rose study subjects is that many of them underwent that diagnostic procedure more than once. And I don't think they suddenly developed endometriosis, that it was missed or they had microscopic disease, very difficult to see. So what we call the definitive diagnostic surgery as a gold standard, it's very, very challenging. And we are kind of stuck using that as the definitive tool. So if a patient undergoes diagnostic surgery and only 1 or 2 biopsies are taken and the surgeon doesn't find endometriosis, is it truly true that the patient doesn't have endometriosis? It's very unclear to us, and we actually don't believe that that few number of biopsies may be enough to confirm that somebody has or doesn't have endometriosis. Another aspect of this gold standard that we refer to as laparoscopic surgery is that the pathologist who views the slides has to find 2 types of cells there. And if they don't find the 2 types of cells, gland cells and stromal cells, if they don't find both cells in the lesion, they say then you don't have endometriosis. And we believe that there are other features of endometriosis that may help in diagnosis, such as fibrosis, that's not included in the diagnostic. And we're hoping through the study of menstrual blood, we can shift the thinking to a more biologically based assessment of endometriosis. If we understand this disease better, we will be able to better detect it as well as treat it. And that's another thing that our lab is really focused on, is developing new treatments. If miraculously we come up with new methods for detecting endometriosis in younger people, I think it's going to be really important to learn more about the treatments and whether they actually halt disease or not.
Mhm.
It's very unclear whether all of the hormone-based treatments actually stop the progression of the disease, and it seems as though they don't in most people. But we've never had a clinical trial that started with people at an early time point to
Mhm.
find out whether taking any of the treatments would change the progression of the disease. And hopefully, with non-invasive diagnostics available, we can have those kinds of studies that are really, really critical to identify the best treatments for the various patients.
Thank you so much for all that information. There's a lot to unpack there, but for time's sake, I'd love to have you come back and discuss for another episode about what those cells are, how they're important, and get into more of the medical and scientific aspects of this very perplexing disease that, like you said, you answer one question and then it opens up another entire plethora of others.
All right. Thank you so much.